All right, thank you, Dr. Lala, for the introduction. Yes, today I'll be talking the brassinosteroid and the muscle function. Very happy to be here, and I'll be able to continue of my work from my PhD at Rutgers, so I'm very excited to present this data today. I'll give a little bit of background of my career, a little bit review on brassinosteroids, and I'll go into the data and explain a little bit of the modulation of muscle loss, and a little update on the lab, the Esposito lab, and we'll cover probably everything that I'm doing these last 2.5 years. So I'm originally from Brazil. I'm from Minas Gerais, a state that is between Rio and São Paulo. I went to attend the Federal University of Viçosa, which is also a land-grant institution. And my first advisor was Professor Murilo Zerbini. He's actually a virologist. I came to Rutgers as an undergraduate student, and I did an exchange program called CAEP, which is an international program throughout I think right now it's 12 countries. But it's an exchange program between my university and Rutgers. And I was a visiting scientist for almost 18 months there. And that's when I was offered the opportunity to do my PhD at Ilya Raskin's lab. So I got my PhD in cell and molecular plant biology at Rutgers, and I got my MBA degree as well at Rutgers. So then I did my postdoc here. I joined NC State in 2013. I have worked in different roles here and positions, but I really want to focus today is since I became assistant professor. So this is the first tour that the dean took us, I think maybe a little bit over 2 years. We went through almost 3 days tour throughout the state, and we could see like the greenhouse and apple production and many things that around us that I was living here maybe for more than 2 years and didn't know. So it was an excellent opportunity. And it was a very good opportunity to see how my research will never have so many happy people taking pictures and being so excited to see a little visiting a farm. So today, I'm just gonna present a little bit of my research, but I want to say I have 5% teaching appointment, 5% extension, and 90% research. My home department is the animal science department, and I'm in the regenerative position. And all right, brassinosteroids was discovered in 1979 with 400 pounds of pollen from Brassica napus, and they isolate 1 milligram of the pure compound Brassinolide. So after this isolation and identification, in this past 40 years, this plant growth hormone have been characterized and used widely in agriculture, has some antiviral and anti-cancer activity reported. They are present throughout the plant kingdom, so you can find brassinosteroids in very low quantity in pollen, but also in seeds, shoots, and in the entire plant. As I said, they are hormones, so they are present in very low quantities. So here is just some examples. I did focus my research mostly in one group, a group of the family of the Brassicaceae. So here is the Brassica campestris, as you can see. The Brassicaceae family is a very interesting family. It's the common broccoli, Brussels sprout, cabbage, cauliflower. Brassinosteroids are very similar to other animal steroids. So here, just to give an example, is a A, B, C, D fused ring with a side chain. And this is the major compound, the Brassinolide, discovered in 1979. But since the discovery of this group of compounds, more than 50 compounds being identified or isolated. So this is a recent publication that shows the characterization and detection in plant material of more than 50 brassinosteroids. And the mechanism, really how it works in plants, was recently discovered. So many years people are after to understand why the regulation is not just in growth, but also in immunity. Why you have a plant that can be a group of compounds like a steroid that can regulate and have a crosstalk with so many other hormones in plants to prevent, to increase yield, but also to prevent some secondary pathogens. So I put here just a little bit of horticulture use. I think was very interesting last seminar series we have when we present about a guest speaker invited by Penny present on stevia. So I thought to add a little bit of horticulture use of brassinosteroid. It's not well used or widely used in US, but very well used in India and in Asia. So here is just a recent review on the applications. And as you can see, for example, in peppers, tomato, potato, spinach, and watermelon, you increase yield, so you have higher yield, but also you have higher antioxidant and phenolic content, for example, in peppers. Or in tomato, they increase the number, size, weight of the fruits, but also lycopene and beta-carotene. So I think this is a very useful information because, as I said, it's present throughout the plant kingdom. So this is present, brassinosteroids are present in any plant. So there is a huge agricultural benefit here and a missing opportunity. But I think 10 years ago when I saw this in the news, I thought was what really caught my attention because this natural plant hormone can help plants eliminate pesticide residues. So this is for cucumber production and was the first time that the application of a natural compound could really prevent pesticide accumulation. So a recent review just released on the brassinosteroid class, the 6th class of phytohormones. So now it's widely accepted as a phytohormone and play many crosstalk in a plant. And what we know about brassinosteroid, we know that they play roles in growth, development, and immunity, but we don't know anything in mammals. If you have consumption of brassinosteroid, what will be the effect in a human or in an animal? So they are structurally similar to animals and insect hormones, and relatively high amounts are found in the Brassicaceae family. Here, just to give an example, this is the Arabidopsis plant, many of you know, and this is a mutant. So in 1998, they really discovered the mutation, but not the pathway, really how it was regulated. So since that, here you can see if you apply exogenous brassinolide on the plant, you can rescue the growth of the mutant. So with that, I started my project maybe 12 years ago at Rutgers, and I did my PhD in pharmacological characterization of brassinosteroid. It was a project directly sponsored by Phytomedics. I received an award, the Eileen Brand Graduate Research Award. It's a recognition of my innovation, of my project, and was patented, and the patent belongs to Rutgers. So we know from this previous study, everything's published, so I will not go through much detail, but homobrassinolide stimulate protein synthesis. What you see here is a dose dependence of homobrassinolide, IGF-1 as a positive control. So you label tritium, so phenylalanine, and you incorporate and calculate percentage of protein incorporation. And as you can see here in the dose of 0.3 micromole to 20 micromole, you really increase protein synthesis and in a dose and a time-dependent manner. I also discovered that brassinosteroid has a huge implication in wound healing. So as you can see here, this is a wound healing mouse model, Excision model, and by day 4, you already have almost completely closure, and by day 8, you have totally closure. But today I'll focus mostly on the muscle function of brassinosteroid. So as I said, we know that has promote protein synthesis and prevent protein degradation. And we know the pathway, how it's doing. It's doing through the AKT, so you have phosphorylation of AKT, and it's also a dose and time dependent. So we decided to move to an animal model, a regular rodent model, rats, and feed the animals for 28 days. And I did a DEXA analysis. So we did the body composition using a DEXA scan. And we have animals treated with 20 milligrams, 60 milligrams, and regular health controls. And we orally gavage them. So what we saw, the animals gain lean, and that lean mass also was converted to, we could see, for example, gastrocnemial muscle have a huge increase. We saw some increase on the bone mineral content, but wasn't statistically significant for the 24 days period. So we know that it's the fiber types that being switched. So we have increase in the type of fiber. So what you see here is just a staining for the type of fiber. So dark is type 1 fiber, light is a moderate type 2A, and darker is type 2B. And what we saw is we have a very big switch and increasing in type 2A and type 2B in numbers, so we have more fibers. So that's how they are really gaining muscle, is the switching of this muscle, of this type of fibers. So as I said, type of fiber 1 is considered for this low contraction, like long distance running. Type 2A, moderate or fast, and type 2B, very fast. So we are focused mostly in the type 2A and type 2B. The pathway, the regulation of protein synthesis and degradation. So we don't know how brassinosteroids really get inside of the cells. They do not bind to the androgen receptor. They do phosphorylate AKT, and they have effect in the downstream signal through the mTOR pathway. So through this pathway, you are increasing protein synthesis and decreasing protein degradation. So when I came here, I decided I have a collaborator, Professor Greg Henderson, who offered me to continue this project, and he had a very interesting idea to do that in an aging model. So I started to have interest in the aging process, and I have a family history of people aging and living more than 100 years, and that really drove my decision to focus on the aging process. So sarcopenia is defined as low muscle strain, low muscle quantity and quality, and low physical performance. It's quite difficult to really characterize sarcopenia because there is no consensus in the definition of the disease. But it's mainly mobility disorders leading to falls and fractures, cardiovascular and respiratory disease, cognitive impairment, loss of independence, lower quality of life, hospitalization, need for long-term care, and death. So I don't think there is any surprise here. It's probably any aging process that will happen to any of us. The main strategy suggested to improve muscle strength is really to gain strength in the early life stage, maintain, and minimize the loss throughout the older lifestyle. So if you are below this threshold, you are in the disability. So if you don't have a minimal strength, muscle strength, you are in the disability threshold. If you have like a low physical performance, sedentarism, for example, you are here in this low physical range, but you still have some muscle strength. And as you see, age for sure is when you're going to have this decline. So you try to prevent or delay is really to maintain throughout your life, but gain in early stages and maintain through adulthood. Sarcopenia cost, indirect medical costs, approximately 18.5 billion in 2000. They don't have a newest release in terms of cost because there is no consensus how really to classify the prevalence of the disease. So I said aging, you have two factors here causing sarcopenia. One is natural aging, and the second is disease, inactivity, and poor nutrition. So they are all related. So as age-associated muscle loss, but you can have inflammatory conditions, which today I'll not be focused so much, but that's where my lab also is focusing the inflammation in so many disease conditions, but also inactivity and malnutrition. So just to give an example here, not to use just a lot of animal models, but a human model here, this is just for you to see, if you take a 25-year-old tide and a 65-year-old tide and do an MRI scan, what you see is a very similar volumetric, but totally different composition of the muscle and infiltration of fat, right? Same you see when you have a lean individual or obese, you do have different size or volumetric measurements, but also a lot more fat infiltration. So I transferred the colony from Rutgers here. It was a very big operation to transfer this colony all the way to this animal facility here. So I'd like to thank so many people who helped in that process. So I bred the colony here. Took almost a year to really establish this protocol, establish the colony here, train people how to do it. But we have a breeding colony. This is sample 8, is the senescence accelerated mouse model. It's called 8 because this is a specific for muscle. There is so many other type of sample. And we decided to do here with, as I said, with the brassicacea. And I chose the cabbage, and I started this project with cabbage seed extract. So we fed the animals with cabbage, 1% cabbage and 1% anthocyanin. Today I will not be focused on the anthocyanin. I'll be focused mostly on the cabbage. And also we have regular diet, low-fat diet, and high-fat diet. And what did we try to measure? Body weight, food intake, endurance. And what we saw is, just to give a little example here, we observe how the animal age. So this is the lean male and the lean female, high-fat male and high-fat female, male cabbage and female cabbage. So one of the things that I haven't assessed prior to this project was if gender specificity for Brassinosteroid activity. So it was first time that I could assess in male and female. And what I'll show here is just the importance of having males and female in designing this type of trials. So here is just male lean cabbage, female lean and cabbage in body weight. Not really big significance difference in 26 weeks. As well in food intake. There is some noise here during some kind of construction in the HMRI, but there is no really statistical significance in body weight and food intake. But there is a difference in the body composition. So the animals, the lean female and the cabbage treated, at 26 weeks, you can see that they gain lean mass. And they do decrease fat. So the question is, if they gain muscle mass, they really convert into function, the endurance. Do they really have more muscle? But that is really just muscle or function. Do they can perform better? So we assess that in a treadmill. And what you see here is 6 weeks, 12 weeks, and 26 weeks. And there's a big difference between male and female performance, but at 12 weeks and 26 weeks, you see that the cabbage-infused treatment improve physical performance. So if they are gaining fat muscle, they are losing fat, they are improving their performance, how's the aging score? So this is our aging SENESCENCE assessment, and what you see, the score is from 0 as a natural behavior, 4 is a not natural behavior. So as the animal age, they move from 0 to 4. 4 is being not reactive, not having any response to escape, or if you try to do anything to the animal, they will be pretty much paralyzed. So what you see, a lower score means they are more responsive. So what you see here, male lean and male cabbage, the male cabbage has a higher responsive to the stimuli, and the female has no significant statistical significance, but also maintains high level of activity. And the passive score from male and cabbage infused, female and female cabbage infused also for this behavioral senescence assessment. So I really do like to do some more assessment, behavioral assessment. We just don't have the capabilities at this point in the animal facility, but I hope in the future, in the next trial, be able to do brain function, cognitive function, and muscle function. But what I can see here is they really gain muscle, they lost fat, they improve performance, and they are aging slower because they are responding much better when you do this behavioral assessment. The mechanism we're still studying. We are not 100% how really is happening, but our hypothesis, Sierra will be presenting her thesis now, April 10. Our hypothesis really, we are modulating the PAX3 and PAX7, which are satellite cells, stem cell markers. So I will not discuss that today. I will leave that for her defense. So brassinosteroid-induced expression of myogenic proteins that lead to increase in muscle mass, increase lean body mass, translate to improvement in physical performance, and is really considered a target for treatment for future approach for repairing damaged muscle. So it's not just for aging process, but athletes, hospitalization, many other processes may benefit from improving muscle. I just would like to give a quick update on the lab. I have so many people working different projects in my lab, not really in the brassinosteroids, so I thought it would be good to just quickly go through and give an update. So I used to do a lot of surgery in the animals, and one of the commitments that I had was to develop new models for in vitro work that could replace in vivo work. So one of the things that I decided, instead of doing surgeries and really doing this excision wound model, we try to do here in the PHHI, we develop using the EVOS flow cytometer, EVOS auto cell imaging, and with this platform of the ORIS, where you have a preventing seeding area of cells. So what you have is like a silicone-based insert, and you insert this in a plate and you prevent the cells to adhere. As you remove the insert, cells will migrate, and as you add fluorescence to the cells, you can detect how many cells and how much fluorescence is detected in that area. So many people are working this project indirectly or benefiting or improving what I develop, and I'm very proud to say that I have many visiting scholars and PhD students that came to my lab and use for many different projects, like in açaí, projects with acerola, projects with hemp, with Alaskan berry, and with soy protein. So I do have to thank a lot of my collaborators. I have a lot of active collaboration searching for new protein sources, but as well for natural sources of brassinosteroids. And I still have active collaboration with John Munafo and my dear professor Tom Gianfagna back at Rutgers. And very happy to have established this collaboration in main campus with Tobias Kaiser and the CMI group. We just recently published this paper, Direct Arachidonic Milk Replacement Trigger Dual Benefit of PGE2, signaling in LPS piglet alveolar macrophage, which my student Caitlin Walter will be defending her PhD next Wednesday, March 20. And I would like to thank also the RCCC, because they partner with me for a very long time, and we have an excellent collaboration. I also want to say my lab is open to new collaborations. That's pretty much what I can offer. I do attend the UGPN group. So this year, we will be hosting the UGPN. So this is a partnership between 4 universities, Surrey University, USP in São Paulo, Ologlon in Australia, and us here in NC State. And April 1st to 3rd, it will be in Raleigh. So if you can, please try to attend. I will present my work in the regenerative medicine in London. This is coming May, and as well in vitro biology. That's a society that I support for more than 10 years. And I'm a guest editor for this special issue that will be released now, March 15, in Frontiers in Pharmacology, Cosmeceuticals from Medicinal Plants. So I would like to invite you for 2 events that is taking place next week and the following in 2 weeks, which is March 20, PhD student Caitlin Walter will present her work, and April 10, Sierra Bon will present her work in main campus. With that, I would like to thank you all for coming. Thank you everybody who could give me support throughout this process, and all my mentors, but special to my daughter. Question?